Welcome. My name is Professor Francis Loth at Northeastern University, and the title of my talk is The Quantitative Structure and Dynamic Nature of Chiari, an Engineering Approach. It is well known that Chiari subjects have cerebellar tonsils that hang lower than for a healthy control. The distance from the McRae line to the tip of the tonsils is referred to as the cerebellar tonsillar position, or CTP. For a Chiari patient, it's typically 5 millimeters or more below the McRae line, whereas for healthy subjects, it would sit above the McRae line. This one-dimensional measurement does not capture the three-dimensional crowding that might be present inside the spinal canal. Here you see here in the axial direction, you see that we see some white, which indicates CSF fluid, and you can see in the sagittal plane it looks very crowded, whereas in the axial plane fluid might be able to get past. So it's difficult to use a one-dimensional measurement to quantify the blockage of CSF flow. And that's why maybe there's been a limited correlation with symptom severity just using the one-dimensional measurement of CTP. In addition, there are many people, approximately 1% of the general population, that have cerebellar tonsillar position greater than 5 millimeter and have no symptoms at all. How is that possible? So we as a group, an engineering group, set out to try and find novel engineering parameters that might help better predict symptomatology, with an underlying hypothesis that biomechanical stress is the cause of damage to neural tissue that leads to symptomatology of Chiari. An outline of my talk is broken down into 4 categories: morphometrics, resistance to CSF motion, compliance in the spinal canal, and brain tissue motion and strain. I'm going to start off with morphometrics. We have studied many different morphological aspects of the brain, spinal canal, and various structures. I'm not going to go through all of them here, but you see a large number of them listed. But I am going to describe the basic overview of the results that we found. Here are the major results. First, the PCF height measurements. We found a general approximately 3 millimeter reduction in many of the vertical measurements, such as the clivus bone, distance to the pons, distance to the bottom of the corpus callosum, vestigium, and PCF. 2, angular measurements showed that a more horizontally angled clivus bone for the Chiari patients, such that this basal angle got larger, also Bugard angle got larger, and the Wackenheim angle got smaller. And then finally we saw a retrograde angulation with a smaller odontoid angle. This generally led to a flattening of the PCF. Next, we looked at cerebellar crowding. I'm not going to go over all the measurements, but we looked at a whole bunch of different things to try and quantify the crowded nature that we see on MR images for Chiari patients. The major results from this study are a larger cerebellum area, that's here number 1 in blue, a smaller anterior PCF area, that's that region 2 in orange, and then smaller CSF spaces anterior and posterior to the spinal cord, down below those two at 3A and 3B. In summary, we found a large number of morphometric parameters that differ significantly between CMI and controls. However, when we examine these morphometric parameters that were different, we find that they're not correlated with the Chiari symptomatology. Still, we have unpublished results that show that anterior CSF space was related to CCOS scores, a measure of surgical success. Now, while the correlation coefficient was modest, 0.4, it was still statistically significant, and thus it might have a utility in predicting to some degree who will be successful from surgical procedure. And then the last thing is that we found age differences in pain became more pronounced with smaller anterior CSF space. Next, I'm going to discuss resistance to CSF motion. Our group has been looking at cerebrospinal fluid motion for a number of years. We like to look at the resistance, the compliance, as well as tissue motion. But here I'm just going to discuss resistance for starters. So this cartoon shows arterial flow coming in red and venous flow coming out in blue. And you have a volume change inside the brain in that gray region. And that volume change pushes fluid in and out of the spinal canal, cerebrospinal fluid. And the difficulty with which to push that out is the resistance. The waveform of that cerebrospinal fluid is pulsatile in nature, and an animation of it is shown there on the left. You can see that the fluid for a healthy case looks very much like a piston going up and down with each cardiac cycle. And there is a significant impact with respiration, but at a lower frequency than the heart rate. Why do we want to look at resistance? Well, one of the reasons is that we've shown that tonsillar position is not a good measure of resistance to CSF motion. So if we do computational fluid dynamics and estimate the resistance inside the spinal canal, we have shown that specific cases such as this one on the right here with a 24 millimeter tonsillar herniation can actually have less resistance than the case on the left with a 7 millimeter tonsillar herniation. And that's because if you look in the axial view, those yellow axial planes, you can see the one on the right has some CSF space around it, whereas the one on the left is pretty much blocked. So we were hoping that the longitudinal impedance will provide a better estimate of the blockage to CSF motion during the cardiac cycle. In this graph here, we see longitudinal impedance for 44 patients, and the ones in blue are for what we call cough-associated headache, and the ones in red are a non-cough-associated headache. And the difference between those two those two headaches is that the cough-associated headache typically goes away 15 to 30 minutes after the coughing, whereas the non-cough associated headache remains. Chiari patients typically present with a cough-associated headache. So we can see that if we compute the resistance between the two groups, we see that there is a definite difference in magnitude of resistance for the cough-associated headaches versus the non-cough-associated headaches. And if we order them from smallest to largest, starting from the center being smallest and coming out largest, we can see that it doesn't really match with the tonsillar position on the bottom graph. So you have some cases where you have large tonsillar position, but maybe not so large resistance, and vice versa. So here's the average data, and you can see that cough-associated headache is like 1,300 versus 300 for non-cough-associated headache. But we still have the cerebellar tonsillar position is larger for the cough-associated headaches, 13.2 on average compared to 9.4. In order to determine how predictive the longitudinal impedance is of someone who's cough-associated headache versus non-cough-associated headache, we plotted the area underneath the curve. And the integral of that comes out to be 0.77 for longitudinal impedance, and 0.7 for tonsillar position. Thus, you could argue that the tonsillar position is not as predictive of those people with cough-associated headache, that symptomatology. However, 0.77 is not particularly good either. We'd rather have a number like 0.9 or 0.95 to really be predictive of a cough-associated headache. Next, we plotted the resistance versus motion. We're trying to understand what the relationship between the tissue motion is with the resistance. On the top two pictures, A and B, you see blue and red columns are longitudinal impedance for Chiari patients in blue and healthy subjects in red. And you can see that the resistance is definitely greater for Chiari patients. The next pane, the middle pane, C and D, that is the maximum displacement in the cerebellum, and E and F is the maximum displacement in the brainstem. And there you see that the maximum displacement coincides with some of the high longitudinal impedance above it, but not all of them. And if you look at the healthy controls, really doesn't seem to make any difference what the impedance was compared to what the motion is going to be. So if we plot that maximum brainstem displacement versus the ILI, you can see that the resistance is correlated with displacement. Correlation values are not huge, but still pretty good, 0.44 and 0.7, showing that there is a correlation between the two. To summarize the CSF motion studies, first off, resistance to CSF motion is significantly larger for CMI cases that have cough-associated headaches, about 4-fold on average. However, there is overlap in the results such that the measure of resistance alone is not strongly predictive of CMI cough-associated headaches, although it was predictive, just not strongly. In addition, resistance is correlated to tissue motion, at least to some degree. Finally, brain tissue motion in these computations was assumed to be rigid, and we could improve these estimates of resistance by incorporating measurements of the brain tissue motion, which we already do with DENS. We have analyzed this previously and shown that brain motion increases impedance by as much as 30%. Although the calculations are significantly more difficult, we feel it's important since we could have cases that with a large motion that might even double the resistance calculation. And then finally, we're interested in understanding how coughing might alter the geometry and in turn the resistance to CSF motion. In other words, looking at the cardiac cycle may not present us with the situations that are those that cause damage to nerve tissue. Maybe it's during Valsalva or coughing, and that the gentle nature of the cardiac cycle is not the real culprit, and we need to look beyond that in order to find situations where damage occurs. Next, I'd like to talk about compliance in the spinal canal. The compliance of the spinal canal is the ability of the spinal canal to change in volume during the cardiac cycle. In other words, as fluid comes from the top, the fluid can exit out the bottom of a segment of the spinal canal, but it can also change the geometry of the spinal canal by enlarging the walls through compliance. So for example, you could have flow come in from the top and not come out the bottom. And if you look at the top few pictures there for the healthy subjects, that's more or less what's happening. If you look at the first healthy one on the left, goes from C2 to C7, you can see that the pulse wave of flow rate has diminished largely by the time it reaches C7. Well, mass cannot be destroyed, so that mass that was coming in through C2 has now increased the volume of the segment between the two slices. And you can see that's true for the other two healthy controls as well, more or less. If you look at the CMI cases, which are on the bottom, there's 4 cases on the bottom, you can see that the waveform was largely maintained from C2 all the way to T1. Even sometimes some of these are beyond C7. Now the magnitude of the pulse is smaller on the bottom slice, but you can see that it maintains quite a bit more of the pulse for the Chiari cases than the healthy controls. So this is an indicator to us that the compliance of the system is changed for Chiari. So these plots look very interesting, but in practice, it's a hard measurement to acquire. The results I just showed you on that last slide were for a 1.5 Tesla scanner, and we could not repeat them for a 3 Tesla scanner for various reasons. So we found also consistency between measurements was a problem. In other words, we had the same subject scanned at multiple different sites, and we couldn't get the same results at each site. So we're trying to understand why this measurement is so difficult to measure, and is there a way we can change protocols to improve that measurement. And one of the things we're working on is this pencil beam measurement technique, which will allow us to do a real-time measurement of the flow at multiple locations. That could be helpful because then we could do things like cough or Valsalva maneuver and try to understand what the compliance is under these more abrupt conditions than just normal cardiac rhythm. All right, next I'd like to talk about brain tissue motion and strain. Working with Dr. John Oshinsky, we've been using the displacement encoding with stimulated echoes, or the acronym DENSE. DENSE is a special MR modality that can measure tissue displacement over a large structure. And the encoding displacements are small, much smaller than the image resolution of the image. For example, the pixel sizes might be 1 millimeter, whereas we've shown that we can measure displacements down to 20 and 30 microns. Remember, a human hair is only 80 microns. So these are really, really small measurements. And these measurements are occurring during the cardiac cycle. So in other words, your brain is moving around very gently in a cyclic manner through each cardiac cycle. And typically healthy people move 100 to 150 microns, whereas people with Chiari are moving 200 to 300 microns. Here we see a couple of pictures. The one on the left is for Chiari and the one on the right is for a healthy control. We get a mapping of the displacement that is occurring across the brainstem. That's the structure on the left, and as well as the cerebellum. And those red hot spots corresponds to the large motion greater than 400 microns. And the blue, the cool spots, are low motion. So the structures on the left, the 2 structures on the left, as I said, were for Chiari, and you have quite a bit of motion, particularly down towards the bottom near the foramen magnum, while the healthy controls has a similar overall pattern, just much lower in magnitude. And you can see the results for a number of patients that we examined. If you look at displacement for Chiari's versus controls, 212 versus 125 for the brainstem, 154 versus 73 for the cerebellum. So it's almost twice. If we put a little circle, that little black circle, so the first one was just an average measurement over the entire structure, but the max displacement is a black circle that we float around to the location with the highest magnitude of motion. So it's sort of like a maximum displacement on that surface. And it's about 10 millimeters in area for that, or one square centimeter, 10 by 10. So if you look at max displacement, it's around 325 versus 156 for the brainstem, and 399 versus 126 for the cerebellum. So even more than twice if you look at a max displacement. So working with Professor Rusbe-Amini at Northeastern University, we were able to compute the strain based on those measurements of displacement for both the brainstem and the cerebellum. And you see here on the top, the Chiari, on the top left is Chiari compression, top right is Chiari extension, and on the bottom, healthy control, compression on the left, extension on the right. And you can quickly see these regions of bright red in the both the compression and extension for the Chiari, but on the cerebellum side. And if you look at the statistics, we show that only the cerebellum had statistically more strain, both compression and extension, compared to healthy controls. The brainstem did not. And that amount of difference was around, not quite double, but something like 75% more compression strain and extension strain in the cerebellum for Chiari patients compared to healthy controls. It should be mentioned, though, that this is a relatively gentle strain. 1% strain is not really considered a large amount of strain. And damage to tissue is considered at more 8 to 10% for neural tissue to have damage. But it's not clear to us that you can get this damage with each cardiac cycle, because these are the strains involved with each cardiac cycle. But maybe we can develop technologies to measure what the strain is during a cough or a Valsalva maneuver. Another test we did was to see if there was a relationship between the stretching of specific nerves and imbalance. So we located regions of specific nerves that are related to imbalance. Those are the vestibular tract, superior cerebellar peduncle, inferior cerebellar peduncle, A, B, and C. And you can see these boxes, a blue box on one side and a red box on the other, and we track those boxes to see if the nerve is getting an overall stretch that might cause it to have damage, and then maybe this would show up on our symptomatology forms for imbalance. So first one I'm showing you here, the top row is strain and the bottom plot is translation. So this one is for the superior cerebellar peduncle. And you can see that there's not a great deal of difference on the top one for strain for the people with imbalance compared to the people without imbalance compared to healthy controls. The same is true for translation. So there was no real difference in this superior cerebellar peduncle. If we go to the next plot, this is the inferior cerebellar peduncle. You can see that there is a few cases with strong strain that maybe tips the average up so that we see a statistical difference, but it's not an overwhelming increase in strain for Chiaris versus controls. There is a difference between with and without, but it doesn't end up being statistically different. And again, translation, a little bit more translation for the Chiari subjects than for the healthy controls. And finally, the strain and translation for the vestibulospinal tract, the VST, with and without imbalance. Here you can definitely see that the strain is greater on the VST for both with and without Chiaris compared to the green controls. Not quite as much difference in terms of translation, but there is not a statistical difference between strain with and without the imbalance. So in summary, for the tissue motion strain studies, brain tissue motion for Chiari is almost twice that of healthy controls for the brainstem and the cerebellum. brain tissue strain is approximately twice that of healthy controls, but only for the cerebellum. The magnitude of the strain is small, approximately 1%. Unclear if this could cause biomechanical damage that would lead to symptoms. Decompression surgery altered brain tissue motion and strain back to nearly the values found for healthy controls. And strain on specific nerve tracts related to imbalance are larger than controls, but no difference is observed with and without imbalance. Currently studying if we can measure strain during a cough using a pencil beam, which is a real-time technique. Overall conclusion. Several engineering parameters show some relationship with symptomatology. Low correlation, but still more than tonsillar position. However, we may require a multifactorial approach that incorporates each parameter rather than looking at them separately. For example, the resistance could be computed using the information of tissue motion to improve the accuracy of resistance to CSF motion. We have shown that that is the case. In addition, patients with crowding may not have significant symptomatology if the subject has a very large compliance in the spinal canal. So that may offset the importance of a high resistance. So somebody may have high resistance, but if they have a large compliance, then the resistance to CSF motion is not a problem because they're able to accommodate that flow through compliance. So therefore, we might need all these parameters to be assessed for a given patient to understand the biomechanics and how they can cause tissue damage. Thank you for your attention.